Genetics is the study of heredity and how genes are passed from generation to generation. The first scientist to be credited with genetics is Gregor Mendel. He suggested paired genes, and he did this by studying pea plants. He studied about 14 different traits of pea plants. And his work summed up to simple dominant traits are controlled by one pair of genes. These pair of genes that we're talking about, alleles. So this is the alternate form of a gene. So there are two alleles or codes for each gene. Each allele will either be dominant or recessive. Dominant alleles are the stronger of the two alleles, and they will always mask the recessive. This is represented by a capital letter, and it is usually written first like a capital T. The recessive allele is the weaker of the two alleles. So this is going to be represented by lowercase letters, and this is going to typically be written second like a lowercase t. A couple of definitions that go along with genetics. Homozygous is also known as pure breeding. This is when you have two of the same alleles for the same gene, or for the gene. So, for example, we can have two capital letters. This would be homozygous dominant, because dominant is capital letters. Or we can have two lowercase letters. This would be homozygous recessive. It's recessive because we have two lowercase letters. You can have heterozygous or hybrid, which is two different alleles. So that's one capital letter and one lowercase letter. And there's a difference between genotype and phenotype. An organism's genotype is the combination of alleles or genes. So it's the combination of the letters, dominant and recessive. And these are some examples. Phenotype is the physical appearance or the physical trait of an organism. So for example, the phenotype of an organism could be tall, short, long, etc. Now we're going to do an example of a Punnett Square. A Punnett Square is a way to organize information when solving genetic problems. So in this problem, we're asked to cross two heterozygous tall pea plants. So the key word here is heterozygous. Heterozygous means two different alleles. And if we're talking about tall pea plants, you typically use the letter of the dominant trait to represent your alleles. So if we're crossing two heterozygous tall pea plants, we're going to represent that with a capital letter and a lowercase letter. You put each letter above each column, and you put the other parent on the left side of the Punnett Square. So now we have two heterozygous tall pea plants. And the way a Punnett Square works, you simply bring over and down your two alleles. So in this square, you have a capital T and a capital T. Over here, we have a capital T and a lowercase t. Here, we have a capital T and a lowercase t. And here, we have two lowercase t's. Remember, the dominant allele is normally written first. So by looking at this Punnett Square, we can see that the offspring produced will be 3/4 tall and 1/4 short. We can also convert that to percentages. So 75% of the offspring will always be tall, and 25% chance that they will be short. A test cross is done to determine the genotype of an unknown organism that shows the dominant trait. So we set up a Punnett Square with a test cross, and the unknown organism must be crossed with a homozygous recessive trait, and you will observe the offspring. If any of the offspring show the recessive trait, the unknown parent has to be heterozygous, because they have to be able to give a recessive trait to the offspring.